a WikiVidi documentaries production. Don't forget to like and subscribe. Enjoy. Neuron. A neuron, also known as a neuron and nerve cell, is an electrically excitable cell that receives, processes, and transmits information through electrical and chemical signals. These signals between neurons occur via specialized connections called synapses. Neurons can connect to each other to form neural pathways and neural circuits. Neurons are the primary components of the central nervous system, which includes the brain and spinal cord, and of the peripheral nervous system, which comprises the autonomic nervous system and the somatic nervous system. There are many types of specialized neurons. Sensory neurons respond to one particular type of stimulus such as touch, sound, or light and all other stimuli affecting the cells of the sensory organs, and converts it into an electrical signal via transduction, which is then sent to the spinal cord or brain. Motor neurons receive signals from the brain and spinal cord to control everything from muscle contractions to glandular output. Interneurons connect neurons to other neurons within the same region of the brain or spinal cord in neural networks. A typical neuron consists of a cell body, dendrites, and an axon. The term neurite is used to describe either a dendrite or an axon, particularly in its undifferentiated stage. Dendrites are thin structures that arise from the cell body, often extending for hundreds of micrometers and branching multiple times, giving rise to a complex, dendritic tree. An axon is a special cellular extension that arises from the cell body at a site called the axon hillock and travels for a distance as far as 1 meter in humans or even more in other species. Most neurons receive signals via the dendrites and send out signals down the axon. Numerous axons are often bundled into fascicles that make up the nerves in the peripheral nervous system. Bundles of axons in the central nervous system are called tracts. The cell body of a neuron frequently gives rise to multiple dendrites, but never to more than one axon. Although the axon may branch hundreds of times before it terminates, at the majority of synapses, signals are sent from the axon of one neuron to a dendrite of another. There are, however, many exceptions to these rules. For example, neurons can lack dendrites, or have no axon, and synapses can connect an axon to another axon or a dendrite to another dendrite. All neurons are electrically excitable due to maintenance of voltage gradients across their membranes by means of metabolically driven ion pumps, which combine with ion channels embedded in the membrane to generate intracellular versus extracellular concentration differences of ions such as sodium, potassium, chloride, and calcium. Changes in the cross-membrane voltage can alter the function of voltage-dependent ion channels. If the voltage changes by a large enough amount, an all or none electrochemical pulse called an action potential is generated, and this change in cross-membrane potential travels rapidly along the cell's axon and activates synaptic connections with other cells when it arrives. In most cases, neurons are generated by neural stem cells during brain development and childhood. Neurons in the adult brain generally do not undergo cell division. Astrocytes are star-shaped glial cells that have also been observed to turn into neurons by virtue of the stem cell characteristic pluripotency. Neurogenesis largely ceases during adulthood in most areas of the brain. However, there is strong evidence for generation of substantial numbers of new neurons in two brain areas, the hippocampus and olfactory bulb. Overview. A neuron is a specialized type of cell found in the bodies of all animals except sponges, and a few other simpler animals. The features that define a neuron are electrical excitability and the presence of synapses, which are complex membrane junctions that transmit signals to other cells. The bodies neurons, plus the glial cells that give them structural and metabolic support, together constitute the nervous system. Invertebrates. The majority of neurons belong to the central nervous system, but some reside in peripheral ganglia, and many sensory neurons are situated in sensory organs such as the retina and cochlea. A typical neuron is divided into three parts: the soma or cell body, dendrites, and axon. The soma is usually compact. The axon and dendrites are filaments that extrude from it. Dendrites typically branch profusely, getting thinner with each branching, and extending their farthest branches a few hundred micrometers from the soma. The axon leaves the soma at a swelling called the axon hillock, and can extend for great distances, giving rise to hundreds of branches. Unlike dendrites, an axon usually maintains the same diameter as it extends. The soma may give rise to numerous dendrites, but never to more than one axon. Synaptic signals from other neurons are received by the soma and dendrites. Signals to other neurons are transmitted by the axon. A typical synapse, then, is a contact between the axon of one neuron and a dendrite or soma of another. Synaptic signals may be excitatory or inhibitory. If the net excitation received by a neuron over a short period of time is large enough, the neuron generates a brief pulse called an action potential, which originates at the soma and propagates rapidly along the axon, activating synapses onto other neurons as it goes. Many neurons fit the foregoing schema in every respect, but there are also exceptions to most parts of it. There are no neurons that lack a soma, but there are neurons that lack dendrites, and others that lack an axon. Furthermore, in addition to the typical axodendritic and axosomatic synapses, there are axoaxonic and dendrodendritic synapses. The key to neural function is the synaptic signaling process, which is partly electrical and partly chemical. The electrical aspect depends on properties of the neuron's membrane. Like all animal cells, the cell body of every neuron is enclosed by a plasma membrane, a bilayer of lipid molecules with many types of protein structures embedded in it. A lipid bilayer is a powerful electrical insulator, but in neurons, many of the protein structures embedded in the membrane are electrically active. These include ion channels that permit electrically charged ions to flow across the membrane, and ion pumps that actively transport ions from one side of the membrane to the other. Most ion channels are permeable only to specific types of ions. Some ion channels are voltage-gated, meaning that they can be switched between open and closed states by altering the voltage difference across the membrane. Others are chemically gated, meaning that they can be switched between open and closed states by interactions with chemicals that diffuse through the extracellular fluid. The interactions between ion channels and ion pumps produce a voltage difference across the membrane, typically a bit less than 1/10 of a volt at baseline. This voltage has two functions. First, it provides a power source for an assortment of voltage-dependent protein machinery that is embedded in the membrane. Second, it provides a basis for electrical signal transmission between different parts of the membrane. Neurons communicate by chemical and electrical synapses in a process known as neurotransmission, also called synaptic transmission. The fundamental process that triggers the release of neurotransmitters is the action potential, a propagating electrical signal that is generated by exploiting the electrically excitable membrane of the neuron. This is also known as a wave of depolarization. Anatomy and histology. Neurons are highly specialized for the processing and transmission of cellular signals. Given their diversity of functions performed in different parts of the nervous system, there is a wide variety in their shape, size, and electrochemical properties. For instance, the soma of a neuron can vary from 4 to 100 micrometers in diameter. The accepted view of the neuron attributes dedicated functions to its various anatomical components. However, dendrites and axons often act in ways contrary to their so-called main function. Axons and dendrites in the central nervous system are typically only about 1 micrometer thick, while some in the peripheral nervous system are much thicker. The soma is usually about 10-25 micrometers in diameter and often is not much larger than the cell nucleus it contains. The longest axon of a human motor neuron can be over a meter long, reaching from the base of the spine to the toes. Sensory neurons can have axons that run from the toes to the posterior column of the spinal cord. Over 1.5 meters in adults. Giraffes have single axons several meters in length running along the entire length of their necks. Much of what is known about axonal function comes from studying the squid giant axon. An ideal experimental preparation, because of its relatively immense size. Fully differentiated neurons are permanently post-mitotic, however. Stem cells present in the adult brain may regenerate functional neurons throughout the life of an organism. Histology and internal structure. Numerous microscopic clumps called nissl bodies are seen when nerve cell bodies are stained with a basophilic dye. These structures consist of rough endoplasmic reticulum and associated ribosomal RNA. Named after German psychiatrist and neuropathologist Franz Nissl, they are involved in protein synthesis, and their prominence can be explained by the fact that nerve cells are very metabolically active. Basophilic dyes such as aniline or hematoxylin highlight negatively charged components, and so bind to the phosphate backbone of the ribosomal RNA. The cell body of a neuron is supported by a complex mesh of structural proteins called neurofilaments, which together with neurotubules are assembled into large neurofibrils. Some neurons also contain pigment granules, such as neuromelanin and lipofuscin, both of which accumulate with age. Other structural proteins that are important for neuronal function are actin and the tubulin of microtubules. Actin is predominantly found at the tips of axons and dendrites during neuronal development. There the actin dynamics can be modulated via an interplay with microtubule. There are different internal structural characteristics between axons and dendrites. Typical axons almost never contain ribosomes, except some in the initial segment. Dendrites contain granular endoplasmic reticulum, or ribosomes, in diminishing amounts as the distance from the cell body increases. Classification. Neurons exist in a number of different shapes and sizes and can be classified by their morphology and function. The anatomist Camillo Golgi grouped neurons into two types. Type I with long axons used to move signals over long distances, and type II with short axons, which can often be confused with dendrites. Type I cells can be further divided by where the cell body or soma is located. The basic morphology of type I neurons, represented by spinal motor neurons, consists of a cell body called the soma and a long thin axon covered by the myelin sheath. Around the cell body is a branching dendritic tree that receives signals from other neurons. The end of the axon has branching terminals that release neurotransmitters into a gap called the synaptic cleft between the terminals and the dendrites of the next neuron. Action on other neurons. A neuron affects other neurons by releasing a neurotransmitter that binds to chemical receptors. The effect upon the postsynaptic neuron is determined not by the presynaptic neuron or by the neurotransmitter, but by the type of receptor that is activated. A neurotransmitter can be thought of as a key, and a receptor as a lock. The same type of key can here be used to open many different types of locks. Receptors can be classified broadly as excitatory, inhibitory, or modulatory. The two most common neurotransmitters in the brain, glutamate and GABA, have actions that are largely consistent. Glutamate acts on several different types of receptors, and have effects that are excitatory at ionotropic receptors and a modulatory effect at metabotropic receptors. Similarly, GABA acts on several different types of receptors, but all of them have effects that are inhibitory. Because of this consistency, it It is common for neuroscientists to simplify the terminology by referring to cells that release glutamate as excitatory neurons, and cells that release GABA as inhibitory neurons. Since over 90% of the neurons in the brain release either glutamate or GABA, these labels encompass the great majority of neurons. There are also other types of neurons that have consistent effects on their targets. For example, excitatory, motor neurons in the spinal cord that release acetylcholine, and inhibitory, spinal neurons that release glycine. The distinction between excitatory and inhibitory neurotransmitters is not absolute, however. Rather, it depends on the class of chemical receptors present on the postsynaptic neuron. In principle, a single neuron, releasing a single neurotransmitter, can have excitatory effects on some targets, inhibitory effects on others, and modulatory effects on others still. For example, photoreceptor cells in the retina constantly release the neurotransmitter glutamate in the absence of light. So-called off-bipolar cells are, like most neurons, excited by the released glutamate. However, neighboring target neurons called on-bipolar cells are instead inhibited by glutamate, because they lack the typical ionotropic glutamate receptors, and instead express a class of inhibitory metabotropic glutamate receptors. When light is present, the photoreceptors cease releasing glutamate, which relieves the on-bipolar cells from inhibition, activating them. This simultaneously removes the excitation from the off-bipolar cells, silencing them. It is possible to identify the type of inhibitory effect a presynaptic neuron will have on a postsynaptic neuron, based on the proteins the presynaptic neuron expresses. Parvalbumin-expressing neurons typically dampen the output signal of the postsynaptic neuron in the visual cortex, whereas somatostatin-expressing neurons typically block dendritic inputs to the postsynaptic neuron. Classification by neurotransmitter production: M.P.A and kainate receptors both function as cation channels permeable to non-cation channels mediating fast excitatory synaptic transmission. MDA receptors are another cation channel that is more permeable to Ca2. The function of NMDA receptors is dependent on glycine receptor binding as a coagonist within a channel pore. NMDA receptors do not function without both ligands present, metabotropic receptors. GPCRs modulate synaptic transmission and postsynaptic excitability. Glutamate can cause excitotoxicity when blood flow to the brain is interrupted, resulting in brain damage. When blood flow is suppressed, glutamate is released from presynaptic neurons, causing NMDA and AMPA receptor activation more so than would normally be the case outside of stress conditions, leading to elevated Ca2 and now entering the postsynaptic neuron and cell damage. Glutamate is synthesized from the amino acid glutamine by the enzyme glutamate synthase. Connectivity: neurons communicate with one another via synapses, where either the axon terminal or an en passant bouton of one cell contacts another neuron's dendrite, soma, or, less commonly, axon. Neurons such as Purkinje cells in the cerebellum can have over 1,000 dendritic branches, making connections with tens of thousands of other cells. Other neurons, such as the magnocellular neurons of the supraoptic nucleus, have only one or two dendrites, each of which receives thousands of synapses. Synapses can be excitatory or inhibitory and either increase or decrease activity in the target neuron, respectively. Some neurons also communicate via electrical synapses, which are direct, electrically conductive junctions between cells, in a chemical synapse. The process of synaptic transmission is as follows. When an action potential reaches the axon terminal, it opens voltage-gated calcium channels, allowing calcium ions to enter the terminal. Calcium causes synaptic vesicles filled with neurotransmitter molecules to fuse with the membrane, releasing their contents into the synaptic cleft. The neurotransmitters diffuse across the synaptic cleft and activate receptors on the postsynaptic neuron. High cytosolic calcium in the axon terminal also triggers mitochondrial calcium uptake, which, in turn, activates mitochondrial energy metabolism to produce ATP to support continuous neurotransmission. An autoapse is a synapse in which a neuron's axon connects to its own dendrites. The human brain has a huge number of synapses. Each of the 1,011 neurons has on average 7,000 synaptic connections to other neurons. It has been estimated that the brain of a 3-year-old child has about 1,015 synapses. This number declines with age, stabilizing by adulthood. Estimates vary for an adult, ranging from 1014 to 5 by 1,014 synapses. Brought to you by WikiVidi documentaries. Would you like to know more?